Hello and welcome to another lecture from Mr. Watkins. This is part 2 of our two-part protein synthesis lecture. Again, I want to remind you that on Canvas the details are in some notes that I placed out there for you. This is just an overview of the process. We started this section of this unit dealing with what the central dogma of biology was, and we discussed the two major parts of how proteins are made, and that's transcription and translation. The first video was on transcription, where the message, the information stored on DNA, is copied into mRNA, and we went over the process of that. Translation is what we're going to go over now. And translation takes that mRNA, that mature piece of mRNA, and basically generates is used to generate a protein. This is done at a ribosome, and translation needs to have a ribosome, and it needs to have tRNA and amino acids, and we're going to get into all of how that stuff works here in just a moment. Okay, you need to remember from previous lectures and previous work in class that a ribosome is made out of protein and rRNA. And remember, this R stands for ribosomal RNA and is made in the nucleolus. Or in prokaryotics, there's another process that, again, you don't need to worry about that where this is generated. So that ribosome is mostly, or the most important part for us, is the rRNA. And here's why. Translation has three steps to it, or we've divided it into three processes. And just like in the other, the transcription, we have initiation as our first step. And the reason why you need to understand about rRNA, as we talk later with evolution, but we need to understand that this is so important for that mRNA to be able to bind to be able to attach to. And so initiation is when that ribosome and that mRNA come together, and that attachment there at the rRNA. And the rRNA on the ribosome and the mRNA, when they interact at the start codon, we say that initiation has begun. Now we need to talk about what a codon is. Can't spell. We need to talk about what a codon is. A codon is a set of 3 nucleic acids that are there in a sequence that code for a specific amino acid. You're going to probably have, if you get a question like this on the test, something that looks similar to what I have here. So if I were to wanted to find, say, what the codon was for proline, PRO is the abbreviation for proline, well, it could be any one of these 4. So the sequence of these codons actually map to more than one amino acid. So let's say that I was given on the test, I needed to know what the sequence was for, or I needed to figure out what amino acid was for the sequence CCA. So here's my first base, there's C. There's my second base, so I know I'm here, I know I'm coming here. There's the second C, and then there's CCA, so we know that that is proline. I also want to point out that there are special sequences that are incredibly important for the process to tell it when to start and when to stop. This code that we have here is pretty much universal for all organisms, from bacteria all the way up to mammals. Okay, so there is a start codon, and you can see that here. That start codon is AUG. And so when we talked about the initiation process, that rRNA and that mRNA are basically trying to come together at that start codon, which is AUG, that's found on the mRNA. Okay, now Once that's found, the correct tRNA needs to come in and bind with that particular molecule. Now, how does that work? Well, first of all, tRNA has to be charged. And what I mean by the tRNA getting charged is energy is basically added, and that tRNA has an amino acid added to it. Okay, so I have a picture of a tRNA molecule here. So here's a tRNA molecule. They tend to be cloverleaf shaped. This is kind of a representation that's typically used to make a tRNA. So remember that the mRNA is going to have a codon sequence, and in this case that codon is GCC. The tRNA, which is still made up of RNA, so these are all the nucleotides that we have here, this tRNA will have what we call the anticodon. Basically, it's the complementary pairs. So that when the tRNA comes into the ribosome, and it will line up and attach to the mRNA at its complement. So big thing to take away, mRNA has that codon, the tRNA has that anticodon. And on the other end, the correct amino acid has been attached. That process by which that amino acid gets attached is called charging the tRNA. So for initiation to occur, what ends up happening is the tRNA is added to that mRNA. Now I'm going to go over something here that is not part or may not be part of your curriculum depending upon if you're AP or IB. Excuse me. The ribosome has 3 spots. It's made out of 2 units, a small subunit and a large subunit. Bring this up a little bit better so you can see it. A small subunit and a large subunit. And right now we're not really worried about what all that means, but we do want to know these 3 spots. And the reason why I bring this up is because it just makes it so much easier to understand the process that's going on. So for initiation, the mRNA comes in, and at this P site here, when AUG is at that P site and is lined up with the rRNA, that tells the process we're going to start converting the mRNA into amino acids. The first sequence, remember the start codon is AUG. So what happens then is the tRNA that carries the anticodon for AUG comes in, and we have our amino acid sitting right out here on the outside. So the tRNA is bonded to the mRNA right here. The next step in the process of translation is called elongation. And elongation is basically the same thing that we talked about before, and that is adding these tRNA molecules to the growing chain, to the mRNA. And again, AP and IB may or may not require you to know this. I know that, but I just know that understanding this process is so much easier if you know these 3 sites. So let's assume that the initial codon has been added. So elongation will occur after the next tRNA gets added to this active site. And once these 2 tRNA are next to each other with their amino acids on the outside, those amino acids are so close together, and there's some enzymes there, that they'll actually form a peptide bond. And when that happens, this whole ribosome shifts down the messenger RNA to the next set of 3, and the new tRNA comes in. And again, there's reference points and things out on Canvas for you, and you can Google it and see it happen and see how people have had it happen. So let's look at this one here real quick for the elongation piece. The P site, I always refer to it as the peptide site, but it's the peptidyl tRNA binding site. Basically, that means a peptide bond is going to form between the amino acids as the chain grows. The A site, I always refer to it as the active site or the accessory site. or accessor, excuse me, site. And the E site is the exit site. So let's walk through how this works. Here's our first tRNA. It's come in, or excuse me, it's probably farther on down the line. Here's our tRNA. It's at the P site. The next tRNA has come in, and so now we have our two tRNAs with our amino acids right next to each other. Because it will have something at the P site, a peptide bond forms. This whole ribosome shifts down 3 nucleotides, the frame. And when that happens, at the E site, the tRNA is released. The the protein that's being formed, these amino acids are released from that tRNA, so it starts growing out of the ribosome. And a new tRNA comes in and starts that process again. The whole thing shifts down 3, and it keeps going and going and going until we get to our final part, which is the termination phase. Sorry, my pencil's running out here. Is termination. And termination is done by, there are codons, there are 3 codons that actually do not code for any amino acid. They are our stop codons, and you can find those on your chart. And so when the mRNA runs through this process and it gets to the end here where it has that stop codon, that stop signal, because the codon, the anti-codon for it, there isn't one for the tRNA for that. And so that signals to stop that we're done. A releasing factor, just another protein, comes in, and this whole process shifts over and releases all the tRNA and that amino acid sequence, which it can go on to be processed, repackaged, and remember that stuff can occur as it goes through the endoplasmic reticulum and in the cytoplasm. Again, that's all the details of this are in Canvas for you so that you can look through and do that. This is just a quick overview to give you some information as you go through. I strongly recommend that you go through and you read through those notes, and there should also be some reference material for you, videos and things out there for you to look at should you desire. That's it. Have a great day.